So here on the Norwich Research Park, the Quadram Institute is at the heart of a community of 3,000 food, health, microbiology and environmental researchers. We're using interdisciplinary research to understand the human microbiome and its influence on health and disease. At the Quadram Institute, we have one of Europe's largest endoscopy units, where research scientists work together with clinicians under one roof. We have a unique pipeline of research, from plants to food, from digestion to health. And this will allow us to accelerate innovation and create new interfaces between food science, gut microbiology, human health and disease. Our aim is to improve individuals' health, in part by harnessing microbiology and genomics. Thousands of different species of bacteria live in the environment around us and within us. We carry at least as many bacterial cells as we carry human cells. If a bacterial species is to be successful, the bacteria need to survive, thrive and grow under diverse environmental conditions. For example, a new bacteria resident of the human gut needs to resist the extremely acidic conditions found upon passage through the stomach, as well as the detergent properties of the fat-solubilizing bile secreted into the small intestine. They also need to compete with many other microorganisms for nutrients under anaerobic conditions in the lower intestinal tract. Another stress bacteria face in the modern era is exposure to antibiotics, which have been developed to treat infections. Antibiotics have revolutionized medicine, but unfortunately, bacteria will always evolve ways to resist their action. Understanding the genetic basis of how bacteria survive and grow in these diverse environments will provide us with opportunities to control them, remove them, or make better use of them. Modern nucleotide sequencing technologies can provide us with huge volumes of DNA sequencing data. However, these sequences alone often provide limited biological meaning, as the function of any genes they encode are not often obvious. Therefore, understanding which of the many bacterial genes are involved in survival and growth in a range of environmental conditions remains problematic. So currently, there's a disconnect between the amount of information we have about the genes, the so-called genotype of the organism, and what these genes do, that is the phenotype. One of the best ways to connect the genotype to the phenotype is to make mutations in genes and observe the resulting impact on behavior of the organism or the phenotype. And this can be carried out under a diverse range of environmental conditions. So, Transposon Mutagenesis is a well-established genetic tool for creating such mutants. Transposons are genes which can insert copies of themselves into locations within target genomes, and they are sometimes referred to as jumping genes. If they insert into a target gene, the chances are that that gene will be disrupted and rendered non-functional. In the laboratory, it is possible to make many transposon mutants simultaneously within bacteria, with each of the mutants having a single transposition event, that is a single transposon, inserted somewhere into the genome. Here at the Quadram Institute, we have developed a powerful version of transposon mutagenesis that allows us to identify the genomic location of the actual insertion site of the transposon inserted into the target genome. This can be carried out simultaneously in what's called a massively parallel experiment. We've called this method transposon-directed insertion site sequencing, or TraDIS, and our more updated version is called TraDIS Express. And this allows us to study the links between genotype and phenotype more robustly. Using TraDIS Express, it is now possible to assay every gene in a target bacterial genome simultaneously, including all the essential genes. And this will allow us to identify all the genes important during survival and growth under a range of diverse conditions, and also to confirm which genes are not involved. This will provide us a much better understanding of bacterial cell biology. The original transposon mutagenesis approaches, like the traditional TraDIS approach, have been limited to only being able to study gene inactivation and therefore only understanding the impact of gene disruption on phenotype. TraDIS Express does this, but it also allows us to study how changing the level of expression of a gene impacts phenotype. This has been accomplished by incorporating an outward-facing, inducible transcriptional promoter at one end of the transposon. Due to the presence of this promoter, transposon insertions upstream of genes can promote gene expression through increased transcription. Downstream insertions that transcribe a gene in the reverse complementary direction can reduce expression of this gene by inhibition of translation due to the presence of antisense RNA transcript. As a result of these additional properties of TraDIS Express, the technology can assay essential genes that the traditional TraDIS methodology cannot assay. For TraDIS Express, a very large transposon mutant collection is constructed. Genomic DNA is then extracted from the entire collection in a single pool and prepared for sequencing by breaking the DNA into fragments and simultaneously attaching so-called sequencing adapters to the broken ends. Then, by performing a polymerase chain reaction, or PCR, using oligonucleotides that hybridize to the sequencing adapter and a second oligonucleotide that hybridizes to the transposon end, DNA fragments containing the transposon end are preferentially amplified. DNA from this PCR reaction, that consists of DNA fragments from all the hundreds of thousands of transposon insertion sites in the mutant collection, are then loaded onto an automated nucleotide sequencing machine. This generates many millions of sequence reads, all originating from the many thousands of transposon insertion sites in this large pooled collection of mutants. Sequence data reads are generated and stored in electronic files many millions of lines long. Within the sequence information is the information we actually require, which is the junction between the transposon insertion site and the gene that's been disrupted. By comparing the nucleotide sequences at the insertion site with a reference genome of known nucleotide sequence, the transposon insertion site can be identified. A sequence read that identifies with the reverse strand indicates that the sequence is derived from a transposon insertion in the reverse orientation. This process is performed for all the millions of sequence reads using computers and bespoke software, and this allows us to identify all the locations where the sequence reads were generated. The transposon insertion site information can thus be displayed against the reference genome sequence, and this is done using vertical bars, with the height of each bar indicating the number of sequence reads that are located to a particular site. In the case of red bars, for the reads are mapped to the forward strand, and the blue bars are for reads that map to the reverse strand, representing transposon insertions in one or other orientation. The relative number of reads at a particular site reflects the relative number of mutants for that site. This then provides a semi-quantitative indication of the relative number of all mutants in the mutant collection. The insertion sites can then be compared with the relative location of the genes in the reference genome. And remember that this resolution of genomic information is generated across the entire bacterial genome. Let's take a closer look at the data. It's possible to identify genes essential for growth under the laboratory conditions used to grow the bacteria. Transposon insertions into essential genes, or those important for growth, will cripple the bacterial cell, leading to the loss of those bacteria from the mutant collection, and therefore the absence of sequence reads generated from transposon insertions, which will largely be absent from these genes. To associate phenotype to genotype, the transposon mutant collection is grown under a selective condition of interest, where some mutants will be more susceptible to the selective growth than others, and therefore some mutants will be more able to survive and grow. Thus, the constitution of the mutants in the collection will change, and these changes are detected using TraDIS Express. When the data are then displayed as before, but this time including the TraDIS Express data from the growth condition of interest for comparison, it can be seen exactly which mutants have changed between the two comparative growth conditions. Mutants with transposon insertions within this gene grow more quickly, and consequently more TraDIS Express sequence reads map to the gene. Elsewhere in the genome, mutants of this gene have been lost from the bacterial population, and consequently sequence reads are largely absent from this gene. In another example, this essential gene lacks transposon insertions and hence lacks any map sequence reads for both growth conditions. However, in the growth condition of interest, there has been a proliferation of mutants with insertions upstream of this essential gene, and the insertions are orientated such that the outward directed promoter in one end of the TraDIS Express transposon promotes transcription of this gene. So this is a great example of how TraDIS Express, using the transposon with the outward-facing promoter, is capable of assaying essential genes when other transposon mutagenesis technologies will fail to do so. And here's another example of the same phenomenon for a gene encoded on the reverse strand. In this section of the genome, there are apparently very few changes in the number of mutants from the growth condition of interest compared to the untreated bacterial strain. But when the same growth conditions are harsher, then obvious differences become apparent. Under these harsher conditions, many of the mutants are killed, but we also observe clusters of genes that become important for growth. Using TraDIS Express, the genes involved in bacterial growth in diverse conditions are quickly identified and can be compared. This vast amount of genomic information provides us with a more fuller understanding of the biology of the bacterial cell, which in turn will help us identify new ways to combat pathogenic bacteria, encourage beneficial commensal bacteria, or optimize bacteria used in medical or industrial processes. TraDIS Express is just one of a number of very powerful cutting-edge technologies being developed at the Quadram Institute. These technologies are promising some very exciting times ahead for our investigation of the human intestinal microbiome, human health, and food safety.